with my another video, and in this video I am going to talk about gram staining, which is the technique to differentiate between gram positive and gram negative bacteria. Please make sure to like, subscribe and share this channel. So, what is the difference between gram positive and gram negative bacteria and why it's called positive and negative? So here you can see the picture in which you can see the gram-positive bacteria in the purple color and gram-negative bacteria in the pink or red color. So they are called positive because they stain positive for the staining, which is called the gram staining, and retains the color purple. And the gram-negative because they don't retain the color purple, and then we have to use another color, and that's why they stain pink and called the gram-negative bacteria. So, why one bacteria can retain the color and the other bacteria cannot? So, there has to be some difference between the positive bacteria and the negative bacteria. So, what is the difference between these two bacteria? So, as I have already said, they are classified by the color they turn in when we do the gram staining. The gram positive bacteria turns purple and the gram negative red. So, here are some examples from the gram positive bacteria like the Clostridium and Mycobacterium, and for the gram-negative bacteria, Proteus and Escherichia coli, which is commonly called as E. coli as well. So, what is the difference between the gram-positive and gram-negative bacteria? So, the major difference that we see is the cell wall. So, in the gram-positive bacteria, which is on my left, the cell wall is very thick. It's 20 to 80 nanometer. But for the gram-negative bacteria, it's very thin, 1.5 to 10 nanometer. And the another major difference, if you see, okay. So if you can see my laser pointer, you will see the peptidoglycan layer in the gram-positive bacteria. So you can see it's a lot. It's many layers of peptidoglycan. But if you go in the gram-negative bacteria, it's very thin. It's one or two layer of peptidoglycan. So this is the major difference between the gram-positive and the gram-negative bacteria. And then we come to the another difference. So if you see in the gram-negative bacteria, there is this outer membrane which is present, but this membrane is not present in the gram-positive bacteria. And all the other proteins like teichoic acid, lipoic acids are present in the gram-positive bacteria, which are important to maintain the structure of the cell wall and also for the growth and the cell division it's required, which are not present in the gram-negative bacteria. And if you see this space, which is called the periplasmic space, the space between the cytoplasmic membrane, this plasma membrane, and the outer membrane, or in this case the peptidoglycan, is called the periplasmic space, which is very small in case of the gram-positive bacteria and big in case of the which is very small in gram-positive bacteria and very large in the gram-negative bacteria. So, how these differences make such a huge difference in the staining, like the purple color and the red color, is what we use to identify different types of bacteria. So as you know, when you get infected with different bacterial culture, we the first thing that we test is to check what kind of bacteria are present, because we need different type of antibiotics for different type of bacteria, because if some antibiotic can enter the cells but other cannot, that depends on the cell structure. So to give you proper antibiotic, we need to know which bacteria are we talking about, and this gram staining is very important in that case. So how do we do gram staining and what is the principle of the gram staining? So as I have already told you, the differences between the cell structure, like the thick peptidoglycan in gram positive and small peptidoglycan cell wall thickness, which is 20 to 80 nanometer for positive and very small for gram negative. So gram staining protocol uses the same features, these features to check which kind of bacteria is present. So suppose you have a mixture of bacteria which is the gram positive, which are mostly cocci shape, means spherical, and the gram negative bacteria, which are mostly rod-shaped bacteria, and you need to differentiate between these two bacteria. So, first we put the crystal violet stain because crystal violet is a positively charged strain and the cell wall components that are in the bacterial cell wall, they are mostly negatively charged. So this positively charged crystal violet can bind to this negatively charged by the electrostatic force or by the ionic interaction. And then we put one fixator which is called iodine. So iodine is a fixator. So what it does is it penetrates the cell and then it forms a complex with the crystal violet using the electrostatic interaction. So it forms a larger complex between the crystal violet and iodine. And now because this all the like the cell wall from both the bacteria positive and negative, it's negatively charged, so the crystal violet can bind there and also iodine can form the interaction between crystal violet and iodine. So now is comes the step where we want to make sure what is what. So we put one ethyl alcohol or acetone, which is a decolorizer. So what it does is ethyl alcohol is a non-polar solvent. So when we treat the bacteria with this decolorizing agent, it will enter the cell wall, and because it's non-polar, it will dehydrate the cell wall, like remove all the water and then shrink the cell structure. And because the larger complex, the crystal violet, which in the gram-positive bacteria, they have a thick peptidoglycan. It will trap this crystal violet and iodine complex in the cell wall. But because in the gram-negative bacteria, the cell wall is not thick, they don't have peptidoglycan, so it cannot retain because you can imagine you have a big circle, you can fix it. But if you have a small circle, and you put 100 people there, you cannot fit all the 100 people in that small circle and then it will collapse, right? Like if you have a small room in which you cannot fit 100 people, and if 100 people come, the room will be filled even outside, and then you cannot accommodate all the people, and then you will see the line of people even outside the door. The same thing happens here. In the gram-positive, because the cell wall is thick and because you have space, it gets trapped here. But because in the gram-negative bacteria, you don't have space, you don't have a thick peptidoglycan, it just loses all the complex of the crystal violet and iodine in the surf, like in the outside, and then it is not stained with anything. So here with decolorizer you can see only the gram positive bacteria are stained purplish, but the gram negative, they just lose all of their color. And then we put another dye which is called saffronine. It's the same dye. It's the same positive. It's also the positively charged dye like crystal violet, and it will also bind to the negatively charged cell component. Because in the gram-positive bacteria, it's already stained with the purple dye crystal violet, so it will go and bind to the gram-negative bacteria and stain this. So in this way, we can differentiate between the gram-negative bacteria and gram-positive bacteria. I hope you like it, and if you have any comments, any question, any doubt, please let me know in the comment section. Please like, share, and subscribe my channel. Thank you.